In this video, I'd like to talk about the composition of blood overall, and then focus a little bit on the plasma and the red blood cells. So the blood is made up of two basic components. There's the fluid component that's called plasma that makes up about 55% of the blood, and then 45% are the cells. Okay, people call them formed elements, and the cells are the red blood cells, which are called the erythrocytes. The white blood cells are the leukocytes, and then the platelets. And so let me switch to a picture that shows something that we're all familiar with. A blood sample is taken, and what you can do is you can put that in a centrifuge and spin it down so that all the heavier elements fall to the bottom of the tube. And this is where the packed cell volume is going to be. And so you can see, what I was mentioning before, that the blood is about 55% fluid. Okay, so that's a little bit over half is plasma. And then the remaining 45% are cells. And here they're saying the 45% are red blood cells, because under 1% of the cells are the white blood cells and the platelets combined. And they always form this little cloud-like layer interface in between the plasma and the red blood cells that people call the buffy coat. And so the buffy coat is just the location of the white blood cells and the platelets once you centrifuge blood down. And so taking a look back at the notes, I want to just go through the basic function of blood, and then we'll look at the plasma a little more closely. So as far as physical characteristics, the pH of blood is not neutral. 7.0 is neutral. It falls between 7.35 and 7.45, which is on the alkaline side. And the temperature is also slightly higher than what you might think, than what body temperature is. So the blood is about 104 degrees Fahrenheit or 38 degrees Celsius. And then as far as an average volume, we do have a finite amount of blood. I mean, that's an important thing to keep in mind. So there's not like an infinite amount of blood. We have this finite amount, and it differs for males and females just based on average differences in body size. So you can see on average we have about 5 liters of blood. And as far as the function of blood goes, transportation or distribution of different items is definitely right at the top of the list, because being a complex multicellular organism, our cells can't go to what they need. So for instance, if we were talking about amoeba or paramecium, they would go to the light or go to the nutrient source. Our cells, being, this multicellular organism, don't have the ability to do that, and so we need some type of transit system to bring the oxygen around, to carry the carbon dioxide that's being generated as a waste product away, to deliver nutrients, to take away waste products, to help with regulation, which I have next on my list, with hormones. And so the blood provides a vehicle to transport items to and from cells, basically. And it's also very important in regulation, because as the blood, and we just talked about this in the last unit, flows through the hypothalamus in particular. Hypothalamus is monitoring our body temperature. Hypothalamus is monitoring our fluid volume and can take action to regulate that if it's outside of a normal range. We also regulate our pH. That's something that we're going to talk about when we talk about the urinary system. So we'll get to that, but the blood's a very important indicator. As you know, that's why people take blood samples and look at the composition and what's in blood and get different characteristics that can give us a clue as far as what's going on in the body. And then protection is another major function with two important roles. Blood clot formation, because we don't have a finite amount of blood. Or, pardon me, we do have a finite amount of blood. We don't have an infinite amount of blood. And so if we spring a leak, you have to plug that up, and that's a blood clot. So we'll talk about that this week. And also white blood cells phagocytize foreign invaders, pathogens. And so that is very protective for us. So overall, these are the functions of blood. And what I'm going to do is I'm going to go through each component of the blood one by one. So in this video, I wanted to talk about plasma and also red blood cells. So plasma is a very watery solution. It's basically water. It's 92% water. And then there are solutes in the water, and the primary solute in the water is proteins. And there are all different kinds of proteins that are found in the blood. Albumin is the protein that's most prevalent in the blood. It's produced by the liver, and actually 90% of plasma proteins are produced by the liver. Another good example that we just looked at in the last unit is fibrinogen, right? So the clotting, or pardon me, we'll talk about that one in this unit. Angiotensinogen. Angiotensinogen we talked about in the endocrine system. And so those are some examples of proteins, and I'm going to switch over to a picture. So I grabbed this off the internet, and you can see that over half of the blood volume is plasma, and plasma is primarily water. And then there are a variety of different proteins that are located there, and then other things like nutrients, waste products, electrolytes, hormones, white blood cells and platelets, and then red blood cells. So going back to the notes, you can see waste products, nutrients, electrolytes, respiratory gases are also in the blood, and those are at much lower percentages than the proteins. So blood is considered a connective tissue. I'll just mention that before I jump into the red blood cells. So the matrix would be the plasma, and the plasma does contain fibers. They're just soluble fibers called fibrinogen. And then the cells are the red blood cells, white blood cells, and platelets. And so it does fall into that category that we talked about last semester. All right, so on to red blood cells. Erythrocytes are red blood cells, and basically you want to think of them as being like a taxi for oxygen. They're just transporting oxygen around. They're delivering oxygen to cells. And so red blood cells can also carry carbon dioxide, just not in, the amounts that they carry oxygen. So we'll look at that. So they have an interesting shape. They're biconcave. They don't have a nucleus. Mature red blood cells don't have a nucleus. They hardly have any organelles. And they're really just highly specialized to transport oxygen. And so oxygen, when it's inside of the red blood cell, what it does is it binds to this molecule called hemoglobin. So let me switch over to a picture of red blood cells. And this is a diagram from our book, which is showing the biconcave disc shape. So all that means is that there's an indentation, like a central indentation on both sides. So that's the two, concave. They're very small cells. This is being measured in microns, so they're tiny cells. And they don't have the typical organelles, like we were saying, no nucleus, no mitochondria. It's really just packed full of hemoglobin. And so hemoglobin is going to fill the cell. And so this shape is really useful because they're kind of like flexible. They can be distorted. And I grabbed some pictures here to show you. So this is an electron micrograph of red blood cells going through a capillary. And you can see how they can kind of like squeeze and distort their way through. And this is an electron micrograph where we can see a red blood cell squeezing through a capillary. And remember, I mentioned that red blood cells are incredibly tiny. And so capillaries are also very, very tiny, so tiny that a red blood cell has to squeeze through. So these cells really kind of take a beating, because all they do is they stay in the circulatory system. So in other words, they're in arteries, arterioles, capillaries, venules, veins, and they're really just transporting oxygen, delivering it to the tissues, and then going back to the lungs, picking up more oxygen, and then delivering oxygen to the tissues. So they have a short lifespan, I guess is where I'm going with that, because they are under such strain and stress in the body. So taking a look at the notes, what I did is I made a little list of the advantages of the structure. So they're flexible. They can stack kind of like dinner plates, if that makes any sense to you. That stack is known as a rouleau. That biconcavity gives a big external surface area to a small internal volume. And so, in other words, remember this cell is shaped a little bit, I'll change that pen. It's shaped a little bit kind of like a donut or a bagel, except without the hole. Instead, it's just going to be an indentation, like we said. And so you can see there's a lot of surface area. This is great because the oxygen that's being carried inside of the cell, all of it is going to be in close proximity to the surface. So we don't want a cell, you know, that looks like a typical cell that we've looked at so far, where oxygen is out here in the central core of the cell. How are we going to, you know, move that out of the cell and drive oxygen into the cell? So in order for oxygen to readily leave the cell and go to a tissue, it's all kind of located on the inside. So that's what I mean by a big external surface area to a small internal volume. All the hemoglobin is going to be close to the surface. And because the cell is filled with hemoglobin, it's dedicated to oxygen transport. And so the function of the red blood cell is that it transports oxygen. And I mentioned that it also transports carbon dioxide. We'll talk about that more in the next unit. Like I said, there's a different way that carbon dioxide travels more than in red blood cells. And because these cells are tiny, we have lots of them. So take a look at this cell count. This is nothing that you have to memorize, but we're looking at millions of cells, 5 million red blood cells in a cubic millimeter. So that's a very small volume. So we have lots of these cells. A male count is different than a female count based on the fact that testosterone drives red blood cell production. And so here we're not comparing the size of the body. Here we're saying if we had an equal volume, right, if we just had this cubic millimeter, that we would see more cells within that tiny space in a male than a female due to testosterone driving red blood cell production. So that's lots of red blood cells. To put that in context, 260 million red blood cell, 260 million red blood cells in a drop of blood. So that's kind of crazy. And I also pulled a picture that I wanted to show you from my PowerPoint. So take a look at this picture. I'm not sure if you can tell what this is. This is an electron micrograph. And here what we're seeing is we're seeing red blood cells clinging to the tip of a seemingly clean hypodermic needle. So hypodermic needle, hypodermic injection is a pretty clean procedure. You know, it's very superficial. It's not a bloody procedure at all. And you can see how dirty the needle is with red blood cells and how small they are, that we would never be able to see that. So the red blood cell is filled with a molecule called hemoglobin. I'm going to move on to that. So hemoglobin is a protein in quaternary structure, which means that it's just made out of 4 separate proteins that then aggregate together. So in other words, these chains, the alpha and beta chains that they're showing us here, I don't mention this in the notes, the names of the chains, you don't have to worry about that. But each one of those, kind of like the variation of orange proteins that you're seeing here, was made as a polypeptide on the ribosome, folded into the shape, and then once all 4 of these were made, they aggregated together. So that's what quaternary structure is. So there's 4 proteins, and then within each of those proteins, there's something called a heme group. So each of those 4 proteins is associated with a heme group, and when you take a look at a heme group, we can see that it contains an atom of iron. That's the Fe. Okay, so that's a single atom of iron, and it's inside of a carbon. I'm going to call it, you know, it's a ring structure, but I call it a carbon frame, if that makes any sense, just for description's sake. So when oxygen binds to hemoglobin, it binds right on the iron atom. So, you know, bullseye. It's easy to remember. That's where the oxygen is going to bind, and then that's where it's going to also dissociate when it leaves hemoglobin and goes into tissue. Okay, so the cells are filled with hemoglobin, and I talked about how much hemoglobin is in a cell. Actually, there's about 280 million molecules of hemoglobin per red blood cell. And then we talked about how many red blood cells we have. We were saying 260 million red blood cells in a drop of blood, or 500 million cells in a cubic millimeter. And with each of those cells having hundreds of millions of molecules of hemoglobin, and each hemoglobin molecule being able to transport up to 4 molecules of oxygen, right? Because each of the iron molecules can transport one oxygen molecule. So that's an amazing, a tremendous oxygen carrying capacity for the body. And it also puts quite a demand on us, because this means that in order to make red blood cells, we're going to have the iron to do that. Okay, so that we're not going to have the dietary intake necessary to keep this going. We're going to have to recycle iron when we get rid of a dead red blood cell. So I think where we should move next, since we've talked about the shape of the red blood cell, the structure of hemoglobin, and the function of the red blood cell, is the life cycle of a red blood cell, let's say. And let's jump over to the notes. So we talked about the structure of hemoglobin. Basically, just remember it as these two components. The heme, being the heme group. We talked about that. The heme group has two parts. There's that organic ring surrounding the iron atom, and oxygen is going to bind to the iron atom. And then the globin is the protein. So as far as the globin, there are 4 globular proteins that link together to form hemoglobin. So oxygen can bind and detach from the iron atom. So the life cycle of a red blood cell, the reason that I'm, kind of going through it like that is, hopefully this makes sense. If we understand, like how a red blood cell is produced, why the red blood cell is produced, what stimulates production of a red blood cell, and being that we're going to have to recycle red blood cells to get that iron, how that's done, that kind of goes through the whole lifespan of a red blood cell, which is very short, about 120 days, about, 4 months. And the reason for that is, like we said, it has no major organelles. It's filled with hemoglobin, and it's just getting pounded down by the circulatory system by circulating around and around. So as far as red blood cell production goes, let's jump over to some pictures, and then I'll come back to the notes and mark these up for us. So taking a look at my PowerPoint, I want to look at a bigger picture first, and then we're going to go down to red blood cell production. So this is a picture that's showing us how there's one cell, there's one precursor cell. It's called a hemocytoblast. It lives inside of the bone marrow, and it gives rise to all of our blood cells, not just red blood cells, but the white blood cells and platelets too. So the hemocytoblast, here you can see it giving rise to white blood cells. Because in our book, what they do is they dissect out the story into the red blood cells, white blood cells, and platelets. But I just want to let you know that the story that I'm going to tell you now about red blood cell production fits right into this story. So that cell, the erythroblast, comes from the hemocytoblast, and it comes from this myeloid stem cell line. And so it would fit right in here. And the same thing with a platelet would be, right in here too. So taking a look at just red blood cell production. So this is called hematopoiesis. That's blood cell production. And when you talk specifically about red blood cell production, people say erythropoiesis. So erythropoiesis is just red blood cell production. So here's the cell that we were just looking at, that I showed you at the very, top of the pyramid, right? That's the ultimate stem cell, the hemocytoblast. That's going to give rise to all of the rest of our blood cells. So the hemocytoblast is going to differentiate through that myeloid stem cell line into basically an erythroblast. So you can see the different stages, pro-erythroblast to an early erythroblast and late erythroblast. And notice that the red blood cell has a nucleus. Now, erythropoiesis, this production of the red blood cells, occurring inside of the bone marrow. So we never see the cell circulating around with the nucleus. Jumping ahead, we can see how the nucleus is going to be ejected from the cell prior to the cell leaving the bone marrow. So the cell is going to transform from an erythroblast, where we make the hemoglobin. So you can see there's ribosomes, there's a nucleus, and transcription translation is occurring. We're making that protein, hemoglobin, so the cell is accumulating hemoglobin, and then it transforms into something called a normoblast. And that's the phase where the nucleus gets ejected and the cell is just filled with the hemoglobin that it produced while it was developing inside of the bone marrow. Once it gets, once it leaves the bone marrow, so it would leave the bone marrow, after we eject the nucleus, it's going to be a reticulocyte. So that is actually a cell type that we would find on a blood draw. So we could actually we could count the number of reticulocytes in a person's circulation, and it would be informative as far as the rate of red blood cell production. So erythrocytes, after circulating for just a few days, will automatically mature into erythrocyte. So that is how red blood cells are produced inside of the bone marrow. And you might wonder, you know, like what is going to stimulate this? Let's take a look at another picture. So red blood cell production is hormonally stimulated. So talking about hormones doesn't end with the endocrine system. We're going to talk about hormones all the way through the end of Unit 6. So using the teeter-totter analogy, which I love, we've used this before, here they're looking at normal blood oxygen levels. If we have hypoxia, so if we have a low oxygen level in the blood, okay, so an imbalance that the oxygen is too low, that's sensed by the kidney. So just as when we talked about the renin-angiotensin mechanism in the endocrine system, how if the kidney sensed a low blood pressure, how it would secrete renin, kidney's also very sensitive to oxygen content. And if the kidney senses there's a low oxygen carrying capacity, what is it going to do? It's going to secrete a hormone called erythropoietin. So kidney would sense that oxygen deprivation, and then it would secrete the hormone erythropoietin, EPO. Erythropoietin is going to target the bone marrow because that's where red blood cell production is taking place. And basically at the level of the hemocytoblast and the myeloid stem cell at the very top of that big pyramid I showed you, erythropoietin is acting to encourage red blood cell production versus other different types of blood cells. So erythropoietin is going to enhance red blood cell production, it's going to increase red blood cell production, and as those red blood cells go out into the circulation, that's going to increase the oxygen carrying capacity of the blood, which should solve our problem. So you might wonder, you know, why would we have a low oxygen level? And there's lots of different reasons, and some of them are just like transient reasons. They come and go. So maybe we have a low red count. Well, this is going to take care of that, right? Because erythropoietin is going to stimulate red blood cell production. Or possibly there's a decreased amount of hemoglobin. Or maybe there's abnormal hemoglobin. And so this could be an issue regarding iron. That's one of the most common reasons that you would see a decreased amount of hemoglobin. Or maybe there's a decreased availability of oxygen. You know, why would there be a decreased availability? Maybe you're at a higher altitude. Maybe you climbed a mountain. Maybe you're exercising and you're breathing fast and you have to deliver oxygen to all of your cells. And so even though there's not a decreased availability, so to speak, if you have an increased demand by skeletal muscle for oxygen, that could feel like a deprivation to the kidney. And so I just wanted to point out there's lots of different possibilities of why and when we feel this deprivation leading to stimulating red blood cell production. So before I leave this slide, what I want to point out is there is no other side of the teeter-totter analogy here. So we always go through the other side, you know, when we talked about the endocrine system. But, you know, if you had a blood oxygen content that was higher than usual, we don't correct for that. So we don't monitor for that and we don't correct for that. Typically, you know, we're going to be 90, you know, in the high 90s% saturated, most likely. And so, you know, if you go to like, you know, 99 or 100 because you're on an oxygen mask or something like that, the hyperbaric chamber, we don't correct for that. So there's no other side of this pyramid. I hope that makes sense. So switching back to the notes, red blood cell production occurs in the bone marrow. Oh, and actually, I didn't write that. I'm glad I'm saying that. So it occurs in the axial skeleton and the proximal epiphyse. So I'm trying to tell you where it's more prevalent, but it's occurring in the bone marrow. Axial skeleton being like the sternum, the ribs, the hip bones, proximal epiphyses, you know, the head of the humerus, head of the femur. So erythropoiesis is part of hematopoiesis. We talked about that. So that's just red blood cell production. And here I listed the cell types for you, and I showed you the picture of that. So we're going to go from the hemocytoblast, which that starts out every story of blood cell production because that's the stem cell precursor to all blood cells. And it goes down the line of erythroblast to form the hemoglobin. Normoblast ejects the nucleus. Reticulocyte enters the circulation. So we would see that in circulation. And then, of course, erythrocytes do the job of transporting the oxygen around. So as far as regulation of erythropoiesis, it's hormonally regulated. And so the stimulus would be the kidney. Oops. I didn't mean to do that. Stimulus is kidney senses oxygen deprivation. And there's lots of different reasons for that. What it will do is secrete the hormone erythropoietin into the blood. It's going to target the bone marrow, the hemocytoblast, encourage it to stimulate red blood cell production because it's producing all of the blood cells, and that's going to increase the number and oxygen carrying capacity of the blood. And so remember, this is going to require some dietary ingredients, iron, B vitamins, amino acids. We would have to have those items in order to make red blood cells. So we also take dead red blood cells, damaged red blood cells out of circulation continually. So we're basically in a continuous equal and opposite like state of production and state of destruction. So red blood cells, we were saying, live about 120 days. And once they're dead or damaged, we take them out of circulation. So we make over 1 million red blood cells per second. And we take that amount out of the blood too. So it's really, you know, quite a process. So I'm going to jump to some pictures here. So here is the production part. This is from our book, where the kidney senses the low oxygen levels, and so it releases erythropoietin, which is a hormone, into the blood. Erythropoietin is going to target the bone marrow and stimulate the bone marrow to put red blood cells out into circulation. So this is a bigger story as far as, you know, how are we going to remove these red blood cells once they're damaged or dead. And so there are two organs that are really good at this, the spleen and the liver. So you can see here they're talking about the liver, and this is going to be a great example because if you have a dead red blood cell, basically what you have is a sac of hemoglobin. So I want you to keep that in mind, even for a living red blood cell. It really is literally just like the sac of hemoglobin. So what you really have to contend with, because this plasma membrane is going to be, you know, nothing for the body to destroy, metabolize, recycle, right? Because all of our cells have plasma membranes, but the hemoglobin, this is what we're really having to pay attention to. So remember, it has two parts. There's the heme group and then those 4 proteins. Well, to get rid of the 4 proteins, that's easy because we can break those proteins down into amino acids, and then you could put those amino acids out into the blood, or you could metabolize the amino acids, and the kidney would take it away as a waste product. So the heme group, this is something different. So remember, the heme group is that frame, the carbon frame, around the iron molecule, or pardon me, the iron atom. And so what we have to do, what the body has to do, it has to break that frame and extract this iron so that we can save that and recycle it. So what we do is, that's what the body does. It's going to metabolize this heme group. It's going to save the iron. You can see that it's stored, it's bound to a carrier, and it's recycled basically. This frame, we metabolize that frame as a waste. And when you metabolize that frame as a waste, what you get ultimately is a bile pigment called bilirubin. And so you might have heard of bilirubin. Bilirubin is blue-greenish pigment. It is a bile pigment, like I said, and it's a product of the liver. But it's a product of the liver as an excretory product. So the liver is producing bilirubin because it's metabolizing this frame. And so bilirubin, it gets put into the bile. So the liver produces bile, Bile has other components to it, but the bilirubin, the bile pigment, it's literally just, part of the dead red blood cell. Now, I know we haven't made it this far in the class, but let me just mention that bile gets secreted into the small intestine. And so when you secrete bile into the small intestine, it does jobs there. But one thing that I want to point out to us today is that basically if you put it in the small intestine, as long as you don't take it back into the blood, then what will happen is it goes through the small intestine, it goes through the large intestine, and then it's excreted as part of the feces. And so we're saying that it's a waste product here, and it actually is a waste product. It will make up a lot of the feces. Actually, the characteristic color of feces is due to this bile pigment. Same thing with urine. The characteristic color of urine is due to some of this does get absorbed back into the blood, and then the kidney filters it out and puts it in the urine. So one way or another, we get rid of this this greenish pigment. So that is how we get rid of red blood cells. Let's jump back to the notes. So once they're damaged or dead, there are these specialized cells in the liver. They're called Kupffer cells. And what they do is they phagocytize, engulf the dead and damaged red blood cells. And then we went through this in the picture. The globin, the 4 proteins, just get broken down into amino acids. And then the heme group gets broken down into the iron atom and the organic ring. So the iron, we bind it to a carrier and we store it and we recycle it. We're going to use it to make new hemoglobin. But the organic ring is going to be metabolized into bilirubin and then secreted into the forming bile. And so bile is secreted by the liver into the small intestine as a waste product, something that we'll talk much more about as time goes on. So once we talk about the digestive system, we'll talk more about that. And then to wrap this up, there are some disorders that we need to know. One is anemia. And anemia, I know you've heard of it. Basically, there are many different types of anemia, but what all anemia has in common is this low oxygen carrying capacity. So the term anemia doesn't really mean like what's wrong. It's always secondary to another condition. It's not a primary condition. And in other words, something is wrong and it's causing this problem of anemia. If we have to fix the primary problem, and then the anemia can go away. But because anemia is, low oxygen carrying capacity, the symptoms make sense. Fatigue, shortness of breath, chills. The 3 main causes of anemia are insufficient numbers of red blood cells. Why would we have, insufficient number of red blood cells? You had a hemorrhage. That's hemorrhagic anemia. So if there's an acute or a chronic, maybe internal blood loss that we're not aware of, and that would trigger us to look for something going on. Hemolytic anemia is when red blood cells rupture in the body. That could be secondary to an exposure to radiation or, something, an exposure to a drug or something like that. Aplastic anemia, that's typically a problem of the bone marrow, that the hemoglobin is not being formed properly in the bone marrow, aplastic anemia. And so that's considered like a precancerous condition. Decreased hemoglobin content, so we're not making enough hemoglobin. The reasons for that are usually dietary. You don't have enough iron. That's usually what people have. Or you don't have enough vitamin D, B12. And that's called pernicious anemia. That's a very hard diagnosis to make sometimes, but that's when you don't have the ingredients to make the hemoglobin. And then another possible reason to have anemia is you have abnormal hemoglobin, and that would be a genetic disorder, something like sickle cell anemia or thalassemia. With sickle cell anemia, let me show you that real quick in our PowerPoint. What happens is it's literally just a point mutation, so it's one amino acid that's different in the protein, in the globin sequence, and what it does is it causes the red blood cell to, kind of like a transformer, to change its shape. It could, like tweak out of shape, and then when it takes on this abnormal sickle shape, it can get stuck in capillaries and be like a dam to blood flow. So it's a very painful condition, genetic condition that we could treat with, transfusions and other types of medications. So jumping back to the notes, polycythemia is the opposite. It's not really the oxygen, but it's when you have an abnormally high red blood cell count. And that's a dangerous condition because it makes the blood too thick. And when the blood's thick, it's very hard on the heart to pump it. And so 3 main types: polycythemia vera, that's a primary genetic condition, that's a mutation. Secondary polycythemia, more common, that would be increased red blood cells due to oxygen deprivation, for instance, smoking. Smoking will cause secondary polycythemia. Person smokes, their kidney feels the oxygen deprivation, puts out the erythropoietin, and all of a sudden you have too many red blood cells. Blood doping is a form of athletic cheating, I guess you could say, where the person takes red blood cells of their own, like banks them, and then before an event transfuses them back, which makes their blood very thick, but it also increases their oxygen carrying capacity, and because of that it can enhance their performance. You can also do blood doping with erythropoietin, which is available as a drug, and there's been many deaths due to blood doping with erythropoietin. So it just stimulates red blood cell production, like I said, enhances athletic performance. So we'll be back and to talk about white blood cells and platelets. Thanks for watching.